Okay, so chapter 18, part 3, this is just talking about erythrocytes. So we're going to look at some characteristics of erythrocytes or red blood cells. We're going to look at hemoglobin and the structure of it and its ability to bind oxygen and carry it. And then we're going to talk about life cycle briefly, and then we'll mention some disorders. So we'll get through the section on red blood cells here. So out of all the formed elements that you find in blood, erythrocytes are the most numerous, with somewhere between 4 and 6 million per microliter. It's going to be different for males and females. Again, it's going to be based on body size in many instances, but that's a rough estimate of a normal volume for red blood cells. Lifespan of a red blood cell is about 120 days. So these are being recycled and rebuilt continuously, just like all of your other formed elements. Functions of erythrocytes, it's largely gas transport. So your oxygen that is coming from the fresh air that's inhaled into your lungs is picked up by blood that's traveling through pulmonary circulation, and it's picked up by the red blood cells. So that's their main job. Red blood cells also do some of the pickup of carbon dioxide from cells as a waste product to be carried back to the lungs to be exchanged that way. But carbon dioxide has a couple of other ways that it is removed from the bloodstream as well. So red blood cells are partially responsible for the removal of carbon dioxide, but almost entirely responsible for the transport of oxygen to the tissues. So we're talking mostly about oxygen transport as a job for erythrocytes. They are what we describe as biconcave in shape, meaning that both sides of the cell are concave or curve inward, as opposed to being a sphere, which would be biconvex. So both sides going externally sort of rounded. They're rounded internally like this. So like a little flat disc with dips in on either side. So it's biconcave. The shape actually lends itself really nicely to the function of erythrocytes. So for the first bullet here that's talking about maximum surface area to volume ratio, when you're talking about the job of a red blood cell to carry oxygen, it's really important that that oxygen can get into the cell and out of the cell really easily, and that's happening through diffusion. When you are talking about diffusion of molecules down a concentration gradient, you may recall this from looking at cellular transport and tonicity back in AP1, but diffusion occurs most efficiently across short distances because, again, you're reliant upon that concentration gradient to power the movement of those molecules. It's passive transport doesn't require ATP. It's just using that concentration difference. And so the shorter distance that you have to travel, the more efficiently that passive transport or diffusion is going to work. So it works out really well to have a lot of surface area on the outside of the red blood cell, but a very small amount of volume on the inside. So diffusion happens efficiently that way. Now imagine that you had a spherical red blood cell. It would be more difficult to get oxygen into and out of that cell because it's got to travel across all of the internal volume of the cell. In the case of a biconcave shape like this, you've got all that external surface area, essentially the same surface area you would have if it were a sphere, only it's shaped like a biconcave disc. So the internal environment or the volume of the cell is much, much smaller than you would see if it were spherical. So it's effective for gas diffusion into and out of the red blood cell. It also allows for flexibility as red blood cells travel through the smallest of your capillaries or your smallest blood vessels. That biconcave shape allows for the cell itself to fold. It's really flexible. So when it gets into those smallest vessels, it has to keep moving through. Sometimes it can fold in half and sort of squeeze through those really, really tiny capillaries. But the blood cells, the red blood cells, are also built with the structural proteins. One in particular that the book mentions is spectrin. And what spectrin does is it allows for that flexibility so that the biconcave red blood cells can fold in on themselves, squeeze through those tiny vessels, and then when they get back into a larger vessel, they can sort of spring back open again. So they're flexible, kind of bouncy. So that's really important for their function in both gas exchange and being able to travel through tiny blood vessels. They are anucleate. Just to review, if you remember in erythropoiesis, one of the steps as you're moving from the hematopoietic stem cells down to your mature red blood cell is that the nucleus is ejected. So the nucleus is lost along the way as the red blood cell matures. So mature red blood cells do not have a nucleus. They're no longer undergoing cell division, so they don't really need one. They're not doing any protein synthesis because they don't have the DNA to do it anymore. They don't have organelles. For the most part, the organelles are lost or deteriorated by the time the red blood cell is mature, which means they also don't have mitochondria. So they aren't using oxygen to make ATP anymore. That's beneficial for a couple of reasons. So first of all, if you have no organelles, if you have ejected or lost all of your organelles, then all of the internal volume of the red blood cell can be used for hemoglobin, which is really important. So by the time you get to a mature red blood cell, you have lost the nucleus, you've lost almost all of your organelles, and you're just left with plasma membrane or cell membrane holding a giant sack of hemoglobin molecules. So we're going to look at hemoglobin on the next slide, and we'll talk about how that works. But hemoglobin is the molecule that's responsible for binding and carrying oxygen. So the more hemoglobin you can squeeze into your cell membrane, the more efficient you are going to be as an oxygen carrying machine, essentially. So the other thing that's interesting about losing your mitochondria means that you can no longer do aerobic respiration, which means that you can't use oxygen to produce ATP. Well, what does that mean for the red blood cell? Does the red blood cell need ATP? Well, it does. It needs a little bit, but they're capable of doing anaerobic respiration, so just simple glycolysis, which doesn't require oxygen. That is good as well because then there's no competition for the oxygen within the cell. So if the cell itself needed oxygen to continue its own metabolic functions, then it would be less efficient at carrying the oxygen to the tissues and letting go of it there. So there's some sort of hypothesizing that that's part of the reason for no aerobic respiration in red blood cells. So really unique cell in the body, really specifically designed, really well suited for its purpose. A really great example of form and function being tightly tied together, as we'll see throughout our systematic review of body organs and tissues. So really cool examples of some of that function going along with form there. All right, let's take a quick look at hemoglobin. When you see the abbreviation HB, like you see here, that's just sort of shorthand for hemoglobin. A hemoglobin molecule is mostly made of protein. They're globin proteins. So you may recall back from the chemistry portion of AP1 when you're looking at organization of biological macromolecules, that proteins have to fold in a very specific stepwise manner in order to reach their final functional form. And you get the primary level of protein organization and the secondary and the tertiary level of protein organization. And you may recall that certain proteins have to reach what we call the quaternary level of protein organization, which means you've got multiple polypeptide chains or protein chains that are interacting with one another in order to be fully functional. So in the case of hemoglobin, you've got 4 polypeptide chains interacting. So you've got that quaternary level of organization with multiple polypeptides. You've got alpha chain 1, alpha chain 2, beta chain 1, beta chain 2. So those are your 4 globin molecules that are going to interact together. And each globin has one molecule of heme bound to it. So heme is this sort of disc-looking structure here in the drawing. And in the middle of heme, you've got iron that is bound there. Heme is also red. So it's going to give, it's a pigment. So it's going to give that hemoglobin its red color. It's going to give blood its red color. It also changes color according to how much oxygen is bound to it. So when we spoke earlier in the first video about the color of blood being able to change, whether it was highly oxygenated or oxygen depleted, that has largely to do with the fact that hemoglobin itself changes color, the heme pigment molecule specifically, where that iron is in the center of it. So you've got globin with heme, 4 of them all working together. Every one of these heme molecules can bind a single molecule of oxygen. Okay, so this is the actual binding site for oxygen. A single red blood cell can contain as many as 300 million hemoglobin molecules. Now, Keep in mind, these are very, very small formed elements. Red blood cells are super small, but they're also incredibly numerous. So when you think about the number of red blood cells that you have, maybe let's say 5 million per microliter, that's quite a huge number of red blood cells, but every single one of them contains 300 million molecules of hemoglobin. And every molecule of hemoglobin, because it contains 4 hemes, can carry 4 oxygens. So 300 million times 4 is 1.2 billion oxygen molecules in a single red blood cell times 5 million red blood cells in a microliter of blood. You're talking about innumerable numbers of oxygen molecules being carried around in your bloodstream to your tissues at any one time, which is pretty impressive when you think about it. Because of this nature of red blood cells, because of the nature of hemoglobin, when you have too few or too many red blood cells or not enough hemoglobin, that concentration is off, you drastically affect the ability of oxygen to be delivered to the tissues. So incredibly important function, oxygen delivery to tissues, right? So that's a little bit more about hemoglobin. Here are the 3 different forms of hemoglobin that you can encounter. And it's really just the same molecule, it just is named for what else is bound to it. So if you've got hemoglobin bound to oxygen, that is oxyhemoglobin. You find oxyhemoglobin as blood is leaving the lungs. It has been freshly recharged with new oxygen molecules, and that heme is bright red. So as we mentioned before, oxygen-rich blood that's on its way back out to the tissues to deliver that oxygen is going to be your brightest red blood, blood in the arteries, blood that's heading back out to, again, deliver that oxygen to the tissues. Once the hemoglobin releases the oxygen that's bound to it, it's going to become deoxyhemoglobin. So just take the oxygen away, deoxygenate it. That deoxygenated hemoglobin is a little bit darker in color. So it's going to be that darker burgundy, almost brownish color that you associate with venous blood that we discussed in the first video too. So that's deoxyhemoglobin. Does it have oxygen or does it not? Now, it's almost never going to happen that a hemoglobin molecule will leave behind all of its oxygen because it's going to be based on sort of need and, again, driven by that concentration gradient. So if there's not a complete depletion of oxygen in the tissues, hemoglobin can hang on to some of those oxygen molecules and sort of carry it around. That's not the biggest deal to understand. What you really want to talk about, being able to sort of remember, is the difference between oxyhemoglobin, which has oxygen bound to it, and deoxyhemoglobin, which has released some or most of its oxygen to the tissues. That difference in color is a good way to differentiate. Also location. So arterial blood, again, is going to have more oxygenated blood in it. It's going to have more oxyhemoglobin, whereas venous blood, which is returning from the tissues back to pulmonary circulation to get oxygen added back into it again, is going to be that darker red color that is associated with deoxyhemoglobin. Okay, and then the third form of hemoglobin, or the third way to describe a hemoglobin molecule, is carbaminohemoglobin, and that is hemoglobin that has picked up some carbon dioxide. Now, about 24% of the carbon dioxide that's produced by cells as waste products is picked up by red blood cells. Again, there are other mechanisms that take care of the other 76% or so of carbon dioxide that's produced. But remember, hemoglobin can pick up some of those carbon dioxide molecules that are being put back into the bloodstream from as a waste product of cellular respiration. So when hemoglobin picks up carbon dioxide, it doesn't bind to the same area. So it does not bind to heme the same way that oxygen does, which is good because that means that the carbon dioxide is not competing with oxygen for a binding site on hemoglobin. So hemoglobin can carry oxygen and carbon dioxide at the same time. So one does not displace the other. But the carbon dioxide instead binds to the amino acids or the polypeptide chains, those 4 globin chains. That's where the carbon dioxide links on, which is why it's called carbaminohemoglobin, because it's the carbon dioxide is binding to the amino acids itself and not the heme molecule in the middle. So those are just some terminology to be familiar with, and it all comes down to what molecules are or are not bound to that hemoglobin at that point in time. All right, so for life cycle of erythrocytes, this is pretty well described in the outline, and it's pretty well described in the blood. So essentially, remember that every 120 days or so, erythrocytes are replaced. Okay, so that's about their life cycle, or their lifespan. So this is a really nice figure. Again, it's found in your textbook. So just read through it, look through it. The idea is basically that the components of erythrocytes are recycled and reused. So you have wandering phagocytic cells called macrocytes, which we haven't gotten to yet, but we'll talk about that in the next section. They're a type of white blood cell. So we'll touch on those there. They go around and they pick up dead, old, worn out red blood cells, and they break down the pieces and they recycle them. So the protein portion, just like any proteins that you ingest through your diet, those proteins are broken down into their component parts, into their amino acids, and those individual amino acids are returned to the nutrient pool to be used to build new proteins. So globin proteins are broken down into their constituent amino acids and recycled. So they might be used to build more globin proteins, or they might be used to build some other type of protein that your cells are synthesizing, but the protein parts essentially are broken down and reused. The heme is going to be broken down as well into waste products, first called biliverdin, which is green, and then eventually bilirubin, which is yellow. So some of those waste products you can see along the way. Biliverdin is the pigment that gives bruises kind of a green color when blood cells are destroyed because of blunt trauma under the skin, and you see a bruise forming. Sometimes you get the weird sort of blue and purples, but you also get that yellow-green color. That's a result of biliverdin, which is a breakdown product of the heme. And then bilirubin is made into bile by the liver. So bile is used to break down fats. We'll talk about that when we get to the digestive system. But a buildup of bilirubin, if you have liver dysfunction, is why people sort of appear yellowish in the skin, which we describe as jaundice. So if you've ever heard of jaundice or known anyone who had that condition, that's why you appear yellow, because there's this yellow pigment that heme breaks down to as it's being recycled. So if you guys have questions about the life cycle of erythrocytes, don't hesitate to reach out and we can discuss it. But I think it's pretty well described in the book and in the outline. So just make sure that you're familiar with the life cycle of erythrocytes. And then to end this section, we're just going to touch on a few disorders of erythrocytes. There are so many different types of anemia that it gets its own slide. And even on this slide, it doesn't list all the different types of anemia that you can possibly encounter in patients. So the term anemia is just a generalized term that means you have deficient red blood cells or deficient hemoglobin. So there's something about your red blood cells that is not functioning properly. There's not enough. And there's a couple of different types that I've listed here for you. So if you see it on the slides and in the outline and in the book, that's a good indication that you probably need to know that type. Or that you may see, excuse me, you may see questions about those types of anemia on the exam. So you can have hemorrhagic anemia. That just means that you've lost blood. So it can happen from injury, it can happen from trauma, it can happen after childbirth, it can even happen with heavy menstrual flow. So it just means that you have lost blood, and that's hemorrhagic anemia. There are plenty of other examples of anemia that are caused by or result from faulty production of red blood cells, faulty production of hemoglobin, decreased red blood cell production. So a couple of examples of those. Sickle cell anemia is one that you probably have heard of. Most people have heard of it, even if you're not sure what it is. The sickle cell anemia is caused by, it's genetic, and it's caused by insufficient types of hemoglobin being produced. So the hemoglobin that's made is kind of a weird shape, and it causes the characteristic sickle cell of red blood cells. So this image here is showing sickle-shaped red blood cells. And that leads to a decreased ability to carry oxygen in some cases. It can also lead to the tendency for those sickle-shaped cells to get stuck together and sort of clog up in small blood vessels. It leads to all types of different symptoms and problems in those individuals. And it's most common in individuals of African descent. And so that's going to be sickle cell anemia, again, genetic. So So that's where you may have heard of that before. You can have iron deficiency anemia, meaning that you just don't have enough iron to produce sufficient hemoglobin. So your hemoglobin levels are low because you don't have the iron that you need. It can be common just due to dietary insufficiency. So you can see it frequently in children and teens. They're growing really quickly and often don't get enough iron in their diet to support all that growth. It's also not too terribly uncommon to see in people who don't eat animal products, so vegans or vegetarians. The iron that's found in animal products is easier for your body to assimilate or pick up and use than iron from plant sources. Not to say that your body can't use iron from plants, it's just less efficient at doing so. So if you do eat a vegetarian diet or a vegan diet, you just have to be careful to make sure you're getting enough iron and that you don't become anemic. There's another deficiency, and that's in B12, and that's called pernicious anemia. So it's just another dietary deficiency that can cause problems. Aplastic anemia is the last example on here that is a type of anemia caused by decreased red blood cell production. And this, is caused by insufficient stem cells that lead to red blood cells. Aplastic anemia can be genetic, some genetic disorder. Aplastic anemia is also not terribly uncommon in patients who are receiving ionizing radiation for cancer treatments. Certain types of toxins can cause aplastic anemia. Aplastic just means that it doesn't form. And then you've got hemolytic anemia. Hemolysis is the term for splitting of blood cells or destruction of blood cells. So hemolytic anemia is caused by overly excessive destruction of red blood cells. So remember, it's normal for red blood cells to be digested by macrophages, to be broken down and recycled. If that's happening too quickly, or in excess of normal levels, that can lead to hemolytic anemia. And then there are lots of other causes of anemia too. Pregnancy can cause it, chronic alcoholism, celiac disease is another listed cause, certain types of cancer, certain types of autoimmune disease, like rheumatoid arthritis, can also lead to anemia. So lots and lots of different forms, lots of different causes. So just be familiar with a few of those that are listed here, and you should be good to go with those. And then two more disorders of erythrocytes to be familiar with. Polycythemia is kind of the opposite of anemia. It just results from excess red blood cells in circulation. And if you'll recall back from the beginning of the chapter when we talked about the properties of blood and we talked about viscosity, remember viscosity has to do with the ability of a liquid to flow smoothly. So the thicker and less sort of easily flowing a fluid is, the more viscous it is. So if too many red blood cells can increase that viscosity, and it can make it more difficult for blood to flow through your vessels. So you got primary polycythemia, which is going to be genetic usually, or can result from bone marrow cancer. So you're actually producing too many cells because of overactive or cancerous RBC stem cells. And then secondary polycythemia is more frequently transient, so it comes and goes, and it can be something as simple as just being dehydrated. So you don't have enough liquid in your blood. That can cause polycythemia or increased blood cells in ratio to the plasma portion, which is mostly water. So it can be something as simple as that. Sometimes you see polycythemia induced by smoking. Blood doping, like we talked about before, is when you are trying purposefully to increase your number of red blood cells in order to enhance athletic performance using synthetic EPO or other methods that can cause polycythemia. And sometimes it can just be from a blood transfusion. So frequently elements of blood are separated when you give blood, and then the part that you need is transfused. So if you just need packed blood cells, they may just put in blood cells and reserve the plasma for another donation, in which case you can sometimes get too many cells. So that's polycythemia. And then it is not in the textbook, but it is in the outline. So I thought we'd mention briefly malaria. It is a significant cause of death and disease in lots of countries around the world. It's not as big of a problem in the United States because of a concerted effort back in the 1800s to get rid of a particular species of mosquito that transmits malaria. So malaria is caused by a protist parasite in the genus Plasmodium. There are 4 or 5 different species within the genus that can cause malaria. And malaria parasites actually get into your red blood cells and cause problems that way. So because they're mosquito borne and because they're carried by a particular species of mosquito, it's only going to be present in tropical climates, because where we used to have them in southeastern United States, but they, like I said, they had a pretty effective eradication program. Actually, the government agency that was tasked with ridding the U.S. of that particular species of Anopheles mosquito transitioned to the CDC after the eradication of malaria, essentially from the southeastern United States. And so the reason that the CDC is headquartered in Atlanta is partially because it was born out of the organization that was tasked with removing malaria from the US. So that's kind of an interesting side note. But most cases now occur in sub-Saharan Africa. Something around 95% of the world's malaria burden falls in the African region. And then there are other places that have problems as well in tropical climates. So that's an interesting side note, clinical significance for a disorder that affects erythrocytes. So that's going to sum up this section of the chapter. We've got to still cover leukocytes and platelets, and then we'll do one final video on blood typing to help you out with that. So look forward to more videos to come.